So I work on brain cancers and trying to develop new immunotherapies for brain cancers. And the important thing to note is that survival rates have not improved for many years. In fact, the last improvement was back in 2005 with a combination of radiotherapy and a chemotherapy called temozolomide. But to this day, the 5-year survival rate is just 5% for these patients. And so immunotherapies, and notably chimeric antigen receptor or CAR T cell therapies, have emerged as a new treatment option and is one of the focuses in our lab. And these are T cells which are engineered to express a synthetic receptor that's capable of binding to target antigens on the surface of tumor cells. And as I hope you can see from this time-lapse video, when this interaction occurs, there's a potent T cell mediated killing response that follows. But one of the challenges when tackling solid tumors has been that many of the antigens we've identified are also expressed at some low level in healthy tissue, and this poses risks for off-target toxicities occurring. So a question for my PhD has been, how can we safely target the antigens that we've identified on glioblastoma as well as other brain cancers? And so today, just briefly, I want to talk through two interesting technologies. The first are called VIPERs, which are synthetic receptors that are similar to CARs in that if they're expressed in T cells, they can detect antigen on a target cell, and this leads to T cell activation. However, due to the presence of a mutated NS3 protein domain, it acts as a binding pocket for a drug called grazoprevir. And if this drug is present, it can block the activation process. And so by changing or increasing the grazoprevir concentration, we've tested a published VIPER and shown that you can have this tunability of T cell activation or the cytotoxic response. And we can measure this via looking at either killing of tumor cells in co-culture assays, as well as looking at interferon gamma secretion by the T cells themselves. So with these positive results, we proceeded to create a variety of novel VIPER receptors targeting different antigens on glioblastoma, with early results for one of our candidates shown here, which effectively killed a U251 brain tumor model in vitro. And what's important to notice is that when compared to testing in a CAR format, the VIPER scaffold results in slightly slower killing kinetics, but it still reaches the same maximal level. But only here, when we overlay or culture it with grazoprevir, we can completely inhibit this killing response. And collectively, this data suggests that we're reaching a point we'd be able to tune the level of activation that occurs potentially after delivering such a therapy, which is important to mitigate off-target toxicities. So the other receptor that I'm interested in are called snipers. And these are synthetic receptors as well, which we can express in human T cells. Now, these function a bit differently. If they encounter antigen on target cells, instead of immediately driving killing, instead, a transcription factor that's held on the receptor is cleaved off. And this can migrate to the nucleus and drive transgene expression. So this inducible gene is where you might encode a CAR, a cytokine, or a VIPER, for instance, with the idea being that the anti-tumor effect or the cytotoxic effect should hopefully be restricted or contained to the region that antigen was detected in. So using a published SNIPER and testing this in human T cells, we've shown that only after antigen exposure with a tumor cell co-culture do we see this induction of the protein. In my system, it currently encodes TAG-BFP, which is a fluorescent protein, and this is just to make the screening process a bit more simple. And we've also measured that this induction can occur as early as 4 hours after co-culture. So to apply this now to glioblastoma, we've collected a variety of samples from a hospital nearby to identify the antigens that are highly expressed, and we do this via cell surface proteomics. And we've identified a list of antigens, and by having the amino acid sequences of various monoclonal antibodies, we're able to build unique antigen binding domains also predicted to bind to those antigens we're interested in. And this forms the extracellular region of our receptor. The SNIPER scaffolds themselves, from the literature, we know that changing the domains in them can tune the sensitivity and function of the SNIPER. And we predict that this is important because we don't know which SNIPER scaffold will be optimal when trying to target a particular antigen with this system. So at this stage, I've created this library and have used Oxford Nanopore sequencing as quality control to validate that the different domains we're interested in investigating are indeed encoded. And they came out with equal distributions, which was great to see. So this is something I'll test in the future in human T cells. So to conclude, I actually want to illustrate one of the gene circuits we're trying to build for this purpose. We hope that it would incorporate multiple VIPER receptors targeting different antigens that we already know are highly expressed on primary tumor tissue samples. And this is necessary to increase tumor coverage for our therapy, but also from a clinical perspective to broaden the eligible patient pool that might be predicted to benefit. If you did this with the CAR format, it would be viewed as quite risky. But by using a Viper format, we have this titratable Grazoprevia off switch. And ultimately, it would be the use of an activation sniper to induce the Viper signaling components, which is how we try to restrict the cytotoxic response that would occur to the region the brain tumor is in. Thank you for your attention, and this is my amazing team that supported me for this work.